My name is Dr. Paul Richardson. I'm the clinical program leader and director of clinical research at the Jerome Lipper Myeloma Center at Dana Farber Cancer Institute in Boston, Massachusetts. And I serve as the R.J. Corman Professor of Medicine at Harvard Medical School. At the ASH meeting this year, I think what we've been really excited to see, speaking to specific areas that we're presenting at the meeting, is the success of Mezigdomide, the oral CELMoD, which has shown such great promise, first as a doublet as Mezigdomide, dexamethasone, and now in combination. At last year's ASH, we presented data on the combination of Mezigdomide, dexamethasone, and daratumumab, as well as elotuzumab. And at this meeting, we're presenting data, or updated data, on the combination of Mezigdomide, bortezomib, and dexamethasone, and Mezigdomide, carfilzomib, and dexamethasone. Now, both of these platforms have been very active. We're reporting response rates of over 70% for both, in fact, closer to 80-85% in combination in relapsed refractory patients. So to see this kind of combinatorial effect of Mezigdomide with proteasome inhibition is a very strong rationale for the ongoing Successor-1 and Successor-2 studies, which are comparing Mezigdomide, bortezomib and dex in Successor-1 to the standard of care, which is pomalidomide, bortezomib and dexamethasone in that particular trial. And that's a global study. And then the second trial, Successor-2, is combining Mezigdomide with carfilzomib and dexamethasone compared to the standard of care, which is carfilzomib and dex. And we're very hopeful that both studies will provide a very strong platform ultimately for the full approval of Mezigdomide in combination for patients with relapsed refractory myeloma. I think what's important to share with you, though, is that at this meeting, we have also reported on a very innovative study in which we combined Mezigdomide with other novel agents and their oral agents in the setting of relapsed refractory disease, in which predominantly patients with not only triple-class refractory, but above all, bispecific CAR-T type approaches that unfortunately failed them. So this constitutes an area of exquisite unmet medical need. And obviously we were pleased in our original study to show that Mezigdomide combined with dexamethasone generated response rates in the BCMA-exposed patients of 50%. So this for an all-oral combination was amazing. In this particular trial, we then went a step further and added rationally designed novel oral agents. Now, the two I want to focus on are tazemetostat, which we'll call Taz for short, and the second one is trametinib. Now, both of these drugs are approved and orally bioavailable in different settings. Importantly, they target completely different pathways in myeloma. And on their own, they're not necessarily particularly active against myeloma in themselves. However, what we had was a strong rationale for combining them with Mezigdomide and to see where that landed. The in vitro and preclinical data were really very compelling for both. And in our study, we were able to show that when you combined Mezigdomide with either Taz or with trametinib, the combination was very safe and well tolerated, which for an oral regimen is very important. But then remarkably, we saw about a 60% response rate combining Mezigdomide and dexamethasone with tazemetostat. But then the real sort of pièce de la résistance was the combination of Mezigdomide, dexamethasone with trametinib. And their response rates of 75% were overall reported, and in some cohorts in excess of 80%. So we were really pleased with this. And these combinations proved safe, with manageable side effects, and very active in patients really in whom all other available classes of therapy have failed them. So a very important step in my view. And we have to recognize that these are relatively small numbers, but I nonetheless think, Cindy, they point to the value of Mezigdomide dexamethasone as a real backbone for relapsed refractory patients to which other drugs can be added and really be successful. And even for that matter, used just as a doublet on their own. And you may say, how does that fit into the therapeutic paradigm? Well, very importantly, we're realizing with immune therapies that you can't just go from one immune therapy to the next. We're realizing with CAR-T therapy that there are lots of potential challenges, and with bispecifics, there's the real issue of immune exhaustion and infections. So putting that all together, drugs like Mezigdomide provide an opportunity to provide therapeutic windows, if you will, for patients in between these strategies and to dramatically improve outcome, at least potentially. And thus far, all of these data would suggest that that is, really what these agents can potentially do. I think another very important positive about Mezigdomide is obviously it's oral. And these all-oral combinations are increasingly important because they avoid the need, to attend clinic, they minimize hospital times, and above all, they're well tolerated. So that provides real value to frailer populations and to populations of patients who may be more vulnerable for a variety of reasons. And so I'm very excited to see them coming forward and these results being so consistently favorable. I think the other highlight at this meeting has been to see the success of Belantamab Mafodotin in combination with other drugs. And I think the DREAM7 data showing a staggering almost 3-year survival benefit in the randomized trial comparing belantamab, bortezomib, and dexamethasone to daratumumab, bortezomib, and dexamethasone really, I think, was a highlight of this meeting. And I think it shows the real value of an antibody drug conjugate in this setting, and it shows the value of combination approaches. And I think what's so interesting as well is the ocular toxicity has become much more understandable, manageable, and less of an issue, especially as we're now able to dose patients with the antibody drug conjugate every 12 to 14 to even 16 weeks. Greatly convenient for patients on the one hand, it's all outpatient, and above all, if we can preserve efficacy in combination, I think this provides a very exciting new platform for BCMA-targeted therapy without some of the challenges that we've encountered with other approaches, recognizing their value, but recognizing also that bispecifics, CAR-Ts, are not necessarily feasible for some of our patients, in fact, for particularly the older and frailer patient, and therefore a platform such as belantamab could be particularly useful in that setting.